So part of the innate immune system involves killing by using what we call reactive oxygen intermediates. So of course, we saw that in the previous class, we saw that the mechanisms for which innate immunity utilizes. Of course, we talked about phagocytosis, we talked about inflammation in general, and cytotoxicity. Okay, so of course, even as phagocytosis is happening, some of the things that are released in the phagosomes would be reactive oxygen intermediates, and they are going, of course, to ensure that death happens for the invading agents. Ideally, reactive oxygen intermediates can be produced in the granules of phagocytes. Of course, if you talk about even the neutrophils, if you talk about macrophages, they are all going to produce granules that then can undergo reactions to become reactive oxygen intermediates that ultimately will be utilized. So in the essence of phagocytosis, it increases the hexose monophosphate shunt. If you remember when we were talking about biochemistry, we looked at metabolic pathways. One of the metabolic pathways for nucleic acids in the nucleic acid synthesis is the hexose monophosphate shunt, although it's linked, of course, with carbohydrate metabolism, the hexose monophosphate shunt. So although it's not among the major pathways that we looked at, but if you remember nucleic acid metabolism, there is the hexose monophosphate shunt. Most of the precursors that are utilized in nucleic acid synthesis are generated. As they are being generated, there is usually the production of NADPH. NADPH, and of course, a key enzyme that is involved in that pathway is NADPH oxidase. And its work sometimes is transfer electrons, and it can transfer electrons to molecular oxygen, creating what we call a superoxide anion. And this superoxide anion can be converted to hydrogen peroxide with the help of superoxide dismutase. And this, of course, is a radical. We can get many radicals from hydrogen peroxide, including hydroxyl radicals. So hydroxyl radicals is highly potential, of course, it's highly potent. It is microbicidal. It is able to kill any microbe, especially bacteria and viruses. And of course, with the combination of myeloperoxidase, we've seen even in neutrophils, there are two types of neutrophils, the primary one that has myeloperoxidase, and the secondary one does not have the myeloperoxidase. So the combination of hydrogen peroxide, myeloperoxidase, as well as halide ions are able to kill bacteria, they're able to even kill viruses. And that is just as a result of phagocytosis. So that pathway would run so that ultimately radicals are being generated, the hydroxyl, the halides, of course, they are being generated majorly from hydrogen peroxidase and the hydrogen peroxide, sorry, not hydrogen peroxidase, hydrogen peroxide. And so, of course, catalase is able to break down on this, then it's able to generate. Okay, so that is one way, no, not one way, but one of the many ways in which death can happen during phagocytosis. For the main function is phagocytosis, but in phagocytic cells, the phagosomes would have what we are calling reactive oxygen intermediates, RIOs. So they are being generated from the granules. We can also have scenarios where still is phagocytosis happening, but nitric oxide is generated, and it's usually generated from the inducible nitric oxide synthase. This is the enzyme that generates nitric oxide. This can be done, of course, in macrophages and neutrophils. We've seen that these are among the two cells that we are calling cells of the innate immune system that are really phagocytic and play a key role in ensuring that phagocytosis is happening. So during phagocytosis, we can produce nitric oxide, and nitric oxide is effective against microbes, especially those that invade the cytoplasm. We also also talked about this protein called defensin, and we know that defensin is a peptide. We already talked about it in terms of innate immunity in general. So it acts like a disinfectant in the phagosomes, of course, during phagocytosis still, and it targets bacteria, fungi, as well as enveloped viruses. We also have other enzymes such as the cathepsin, the lysozymes, the lactoferrin that are able to kill bacteria, and they usually are hydrolyzed because most of these enzymes are hydrolytic. So they add water components onto the bacterial membranes, and once that happens, then the membranes are compromised, and then of course they become porous, and the bacteria will be killed. So most of these include neutral proteinases. You know what proteinases are? These are enzymes that are able to break down any proteinous components. Maybe if they are rich in a particular amino acid like serine, would be a serine protease. If it's lysine, it would be a lysine protease, and so on. So cathepsin G, of course, is one of them, which is, of course, a serine protease. Lysozymes, lactoferrin, they're able to interfere with the bacterial membranes. They break down the bacteria. They add water components on the membranes of bacteria. And once that happens, then the membranes become porous, and of course, then the bacteria is able to die. Okay, so that is ideally phagocytosis. So in the phagosomes, there's some of these things that are able to kill the invaders, include the reactive oxygen intermediates, nitric oxide, defensins, enzymes. They are all combined in the phagosome, and they're able to eliminate the invader. So of course, the key phagocytic cells would be the macrophages and the neutrophils. So I talked about innate immune cells being three. So far, we've covered two, macrophages and neutrophils. We are yet to cover one. In a moment, I think we're going to address the other one that is remaining. But majorly, these two, and this is killing by phagocytosis. So phagocytosis, we know the entire process, but we have other ways in which the phagocytes are able to then eliminate what is in their phagosomes.